Hello, Houston. Thank you for being here with us today. It's a fantastic meeting so far. We are very excited to be here in Houston, home of NASA. Particularly for me, Texas is a special place because I started my PhD here in Texas, and some of our early IP came from Texas A&M University. I remember, like, as a PhD student going to NASA and being amazed with everything they do, and to come here today as head of R&D biologics and to see NASA presenting that the technology we develop is going around the Earth and going to Moon and Mars is just mind-blowing. So thank you. So we have a very exciting technical update for you today. This is our forward-looking statement. So usually when we present our technical updates, we first go with what we currently do, our product update, before going to R&D. But today we are doing it in reverse order. I will present what we do in R&D now, some of the cool science coming out very soon. And then Rosemary will come and talk to you about our products and timelines. So every year we present our technical updates in two meetings, London Calling and NCM, and all these talks are available online if you want to go and have a look. This is our technology. I'm sure most of you know about this, but thanks to Jim, our 21% of you are apparently newcomers. So for your benefit, so we have a membrane. It's a synthetic membrane. It's very stable. And then within that, we have a biological nanopore. And the nanopore is heavily engineered. Then we have motor protein, shown in purple. This motor protein is also heavily engineered, and it controls the movement of DNA or RNA translocating through the pore. We have other cofactors, magnesium and enzyme fuel. We have run conditions, pH, temperature, and so on. And then we have salt solutions, and we have electrodes above and below the membrane. So we can measure the current passing through the nanopore. When the DNA is translocating through the pore, we can detect the ionic current signal change based on the sequence changes. And we have this set up in every single flow cell that you use, whether it is Min9 or Promethean. In each well, we have this set up, and then we use these flow cells in our platforms, whether it is Min9, GridION, Promethean, or P2. So this is the same setup in both DNA and RNA sequencing. And today, I'm going to talk about RNA first before going into DNA. So RNA is very similar to DNA sequencing, but we have a very specific RNA-specific nanopore. We have a RNA-specific motor protein. And because we do native molecule of RNA sequencing, we don't have reverse transcription bias. So nanopore technology is the only technology you can use to detect sequence native RNA molecules without converting them to cDNA first. Because of our technology, there are a lot of things that you can do now that you couldn't do before. Modification detection is one thing. Like in DNA, we can do RNA modifications, and we can detect poly-A tail very accurately. And we can do complete isoform mapping and transcript quantitation as well. We have recently released RNA-004 kit. It is an upgrade to RNA-002, which was our previous kit. And this new kit is now available in store, and it is in open early access. So with the RNA-004, you get some amazing upgrades. Accuracy is improved significantly. Previously, it was about 92%, and now you get about 96% accuracy with our direct RNA sequencing kit. More importantly, you get very high output. In previous kit, the enzyme runs around about 70 bases per second, but in the new kit, enzyme runs at about 120 bases per second. It's twice speed, which improved output about 3 times. And you can see in this plot on the left that in a single Promethean run experiment, you can get about 35 million reads in RNA. This has been used during the early access program. Many of our collaborators or many of our developers use this kit, and I'm very pleased to say that most of them got the exact outcome that we experienced in R&D. So like in DNA, we can do base modification detection in RNA as well. We have checked about 7 different bases, and every single modification we can see in the signal. But M6A is one of the most important modification in RNA biology. Especially M6A is very much enriched in mammalian RNA, especially in this DRARCH context. So DRARCH is, for those of you who don't know what is DRARCH is, it is something like CPG in DNA. So in the panel at the bottom, you can see the DRARCH sequence context. So we have developed M6A modification detection model. Like in DNA, we used synthetic randomers and used a Remora style model. And with that, you can get about 98.95%, so almost 99% accuracy in M6A. So this is very exciting for RNA biologists. On the right-hand side panel, you can see a metagene plot for HEC293 transcriptome. So this is done in collaboration with IIT Milan, and they gave us some of their biological sample data. So in this, what you can see is how we can detect M6A modification in transcriptome. So in the bottom, you can see the structure of transcript. So you see 5' UTR and the coding region and the 3' UTR. In red, what you see is wild type transcript. So apparently in M6A is heavily enriched in the 3' UTR, and especially at the especially at the stop codon region. So you can see a huge peak at the stop codon, indicating that there's so much M6A modification there. So the middle one in orange is in a condition where the M6A writer enzyme is inhibited. So you can see the less amount of modification. And in green, what you see is the control where there is no M6A modification. It's the synthetic control. So you can see clearly how clearly we can detect M6A modification in transcriptome. M1 pseudouridine is another important modification, especially in mRNA vaccines. All uridine is 100% modified with pseudouridine because it makes the mRNA vaccine molecule more stable. And we have developed a model to detect M1 pseudouridine as well. It's a 4-base model because every single uridine is modified with M1 pseudouridine in mRNA vaccines. So in the bottom panel, you can see different transcripts. And in every transcript, you can see around 95% accuracy, very similar to our direct RNA base calling accuracy. So we are working, we have recently announced a collaboration with BASE. BASE is one of the largest mRNA vaccine manufacturing facility and research facility in Australia, working with the University of Queensland. So they have recently published a paper. They are developing methods, QC methods, to use Nanopore direct RNA sequencing for mRNA vaccine QC. So this is going to be an interesting area to watch. So we have done Dorado update recently. In that Dorado upgrade, there is some interesting features for RNA as well. One of the interesting features is detecting poly-A tail length. So poly-A tail length is in different mRNA molecules. It's different RNA molecules. It's in different size for numerous reasons, but it's very important to detect how long the poly-A tail is for various applications. So in this example, we are showing poly-A tail length between 10 and 100 each bar is showing the exact length of the poly-A, and you can see in each transcript, we can detect very accurately what the length of the poly-A tail is. And this is also true for M1 pseudouridine IVT transcripts, and in the right side panel, you can see even with that M1 pseudouridine modified RNA, you can detect poly-A tail very accurately. In addition to poly-A tail length, this Dorado upgrade also include other features. It improves read splitting, and it also gives better adapter trimming as well. So we have also done some software optimization. So far, when we were with our RNA-002 kit, you couldn't do proper writing with for RNA longer than RNA shorter than 200 bases, mainly because software detected something wrong with that and put that into failed folder. So there are some new software updates in coming along. This is not released yet, but it will come soon. So with that, you can detect RNA molecules all the way down to 50 bases. So this is very important to detect, to be able to sequence short RNA. It is important in tRNA. It is important for Cas9 guide RNA, as well as degraded samples. Short sequences are there. So to be able to sequence that is very important thing in RNA biology. So in this example, what I'm showing is human transcriptome is shredded into about 100 nucleotides. And you can see, compared to the baseline, the optimized software detect it and sequence it, and you can get very high output with that improved software. And also on the right-hand side panel, you can see that the accuracy of the short read is very similar to long read. So accuracy doesn't depend on the read length. So now the summary of the direct RNA. What is available now is RNA-004 kit, and it has improved accuracy at 96%, 3 times more output compared to the previous kit, and we can detect, we can call M6A very accurately to about 99%, and we can also do very accurate poly-A tail length prediction. So in research, we are doing the short RNA sequencing. Hopefully, it will come soon. And we are also working on barcoding multiplexing, because we know that is one of the key features that a lot of you are asking. And we are also working on improving sensitivity and releasing base calling modification calling for M1 pseudouridine. And we are also working on other modifications as well, and targeted RNA sequencing. So now, moving on to DNA. So we have 2 modes of DNA sequencing, Simplex and Duplex. So we have talked about these things extensively. Simplex is what I explained earlier. One strand is going through the pore, other strand is going away. And in Duplex, we try to sequence both strands. So in Simplex, we have improved our accuracies significantly over the years. And we have now Q20 accuracy, Simplex accuracy. And we also get very high output. We get about more than 100 G for 1 Promethean flow cell. We are also improving further modifications in DNA space as well. So as you know that we can do 6MA, 5MC, and 5HMC very accurately. So we are trying to add more and more modifications into DNA space. So in this example, I'm showing is 4MC. 4MC is actually, it's becoming, it's kind of more challenging to get a model, but we are working on it. So in this example, in the top panel, what you see is a control where there is no modification. Dark blue is where the C is. And in the bottom panel, what you see is synthetic strands. And on the top of the bottom panel, you can see where the 4MC modification is. And you can see in a turquoise color all the 4MC modifications that you can detect with the base modified caller that we are developing. It is not released yet. We are still working on to improve that. Hopefully, that will be released soon as well. Not only on modifications, we are also working on DNA damage. So DNA damage is important, especially in cancer biomarkers, DNA damaging. If you can detect DNA damaging, that is very important. So in the panel, what we are showing is detecting 8-oxo-G and ribo-G. So in the blue panel is where the modification is. The blue bar is where the modification is. In red is the modified strand, and the black is the control. And you can see around the purple bar, there is significant changes into the signal between black signal and the red signal, which means we can detect those things in the signal, and we will develop, like modification callers, we will develop callers to detect these DNA damage modifications as well. So going into Duplex, so as I said earlier, so in Duplex, we try to detect both complement and template strand. When the template strand going through the pore, after template strand going through the pore, we also capture the complement strand, and then we get the two sequences together, and we give you the duplex accuracy. So because we are compiling the two together, the duplex accuracy is much better than simplex accuracy. So simplex is Q20, and with duplex, you get about Q30 accuracy. So in the panel, you can see at the bottom one is R9 chemistry, which is our previous chemistry. Some of you are still using it, but we are trying to move everything to R10 chemistry. So in R10 simplex, you can see less errors compared to R9, but in R10 duplex, errors are significantly reduced. I highlight again that in duplex, we get Q30 accuracy. And with duplex, you can do quite a lot of interesting things that you cannot do with any other technology. And one application is hemimethylation and detecting asymmetric methylation. So in the left-hand panel, you can see CpG site. And if you take 5MC and 5HMC, there are 9 different permutations that you can detect. So in the middle panel, you can see that we can detect these differently asymmetrically modified CpG sites. And in the right-hand panel, you can see that we can also separate that into different haplotype as well. So this is something only nanopore sequencing can do, and only duplex sequencing can do, and it is not possible to do this with any other technology. And this is now available in ModKit. So ModKit is in the newly released Dorado as a new feature as well. So with duplex, what we can do. So when Karen Miga and the telomere to telomere consortium filled the gap of the human genome, it was very interesting to see. And in that particular project, they used multiple different technologies to do the assembly. In nanopore sequencing, being ultra-long DNA sequencing technology is one of the technologies they used. Since then, we have been developing methods to use nanopore-only telomere to telomere assemblies. So we have done that, and many scientists, so we have shown that it is possible to do nanopore-only telomere to telomere assembly. As you can see on the left-hand panel, it's a full chromosome assembly. And on the right-hand side, you can see that chromosomes are separated, and assembly accuracy is Q42, and we have N50 on 135 MB. So we are still working on to improve accuracy further, and especially to improve the consistency of the accuracy. So when we improve the consistency and the accuracy, so we can change multiple components. You can change sequencing chemistry. You can change sample prep. You can change run conditions, and also improvements coming from platform side. And software plays a much bigger role in improving accuracy as well. So generally, when we improve accuracy, it's not just one thing. It's a kind of multiple of different things that we bring together to improve the accuracy. So recently, we've been working with machine learning teams, and we've been engineering enzymes using machine learning approaches. So in this panel, so we are using protein language models, 3D structure prediction, hybrid models. And when we kind of improve enzyme, it's not only just how the enzyme functions with DNA, but it also matters how enzymes sit on top of the pore. So we have to balance the interaction between the enzyme and the pore as well. So while we are a machine learning team developing approaches to design thousands of new mutants, our chemistry teams were also developing high throughput protein purification methods from lysate to sequence library, and we were also developing very high throughput analysis method. And to put the two together, so we can now purify sequence thousands of mutants very quickly. And in this plot, what we are showing is some of the results. So each dot in this plot is a different enzyme mutant. In the middle, the dark blue dot is our baseline enzyme. So above the horizontal line, so the x-axis is the relative speed, y-axis is the relative accuracy. So above the horizontal line, what you have is better enzymes with better accuracy. And right side of the vertical line, what you have is enzyme mutants with better speed. And generally, the speed and the accuracy don't go together. When you try to improve the speed, accuracy goes down and vice versa. So it's very delicate balance between the two to get this highly accurate enzyme with better speed. So we have picked few enzymes, and one of the enzymes that we picked is becoming important. So this enzyme mutation, I call it E8.2.1. The reason we call it E8.2.1 is the enzyme in our current kit 14, is called E8.2. So this version, new version, is a very minor change. It's nothing really, nothing much changing at all, actually. So you can see at the bottom the signal between the E8.2 and E8.2.1 is almost identical. There's not much change. However, this mutation does improve accuracy. So on the panel on the right-hand side, you can see that we have checked this particular enzyme mutation in different conditions. So each dot in the accuracy plot is a different condition. Towards the bottom, you have different platforms from fungal to Minna and towards the Promethean. So we have tested different sample types and different temperatures and so on. So what we see is with this mutation, accuracy is consistently better than E8.2, even like 1 or 2 Q points. And also, at the bottom panel, you can see the output graph. So none of these conditions change the output. So output, usually when we improve the accuracy, output goes down. But in this particular case, output doesn't change at all. So we have picked up this mutation because it doesn't change the signal. So it's almost the same as E8.2, which is our current enzyme. And it improves accuracy. So, and with this, we have also, now when we change enzyme, we also have to be careful because it has to also base call, not only just give the general accuracy, but it also need to give similar or better modification calling as well. So we can't introduce anything with lower modification calling. So we have checked modifications as well with 5MC, 5HMC, and 6MA. For 5MC and 5HMC, it didn't change the accuracy at all. It's almost the same. But with 6MA, actually, it improved 6MA accuracy as well from 96.3% to 97.7%. So this is good news for us. It improves accuracy. It improves modification accuracy. It doesn't change the output. So when we went ahead and tried to use this enzyme, and so general accuracy of our current kit, E8.2, which is in kit 14, with the, what you currently see is version 4.2 in Dorado, and with that you get Q22 accuracy. So we have released a new version of Dorado yesterday with a new model. So the Dorado version is 0.5, and with that, we also have released a new model for your current, for our current Kit 14. And with that, you get Q23 accuracy. And with that same model, if you use E8.2.1, this new Enzyme version, you get Q24 accuracy. So we go from Q22 to Q24 accuracy. This is significant because we know even though we say you get Q20 with our R10.4.1 Kit 14, some of you don't get that, some of you get Q18, Q19. And we believe that with this particular change, most of you will be able to get above Q20 line. So we are saying Q20 consistently, accuracy, consistently you should be able to get Q20 with this particular change. And because it is, as I said, it is a minor change, it doesn't change much other than improving accuracy. And we hope that we can change this enzyme seamlessly into existing kit without making a big noise about it. So that will happen in Q1 2024. So this new enzyme will go into our kit seamlessly, and you will see much better accuracy consistently with ONT kits. So what we can do more with it, we have, while we are kind of planning to release that in Q1 2024 as existing kits, we were also, in research, we were also trying to improve accuracy further with this enzyme. So we have checked many different conditions. So in this one particular condition, we saw that accuracy improved beyond Q24. So our machine learning team was also developing new next generation of base callers. And this next generation of base callers with new models, even though they are new models and base callers, they can seamlessly go into Dorado as well. So again, not a big change. But with that, we will be able to hit Q28. So this is a significant improvement to our Simplex accuracy, going from Q20 to Q28. So hopefully, this is, I want to highlight that this condition is still very much in research. We will improve it even further. What we want to do is hit Q30 accuracy in Simplex. But we were trying to see what we can do with this new highly improved Simplex accuracy. So one thing that we can do is try to see whether we can do T2T ONT only assemblies with this particular condition. So I'm not an assembly expert, but we have one of those here today. So I'd like to invite Sean McKenzie. He's our Associate Director of Genomic Applications Bioinformatics. So he will talk to you about what we can do with this high accuracy Q28 Simplex data.
Hello, everyone. Now, I don't think I need to convince this crowd that telomere to telomere genomes have been an amazing advancement in genomic science, finally allowing us to capture the complete range of genomic variation in a given sample. This has implications for personalized medicine, genomic diagnostics, and basic genome structure and function research. Now, despite these advantages, it still remains somewhat niche because it is still somewhat challenging to generate T2T assemblies. For one thing, it requires at least 3 different data types right now, as we've talked about multiple times. You need some near-perfect long reads. For instance, our Duplex reads have been the way we've accomplished this in the past. You need ultra-long reads, and you need some form of long-range phasing information, for instance, POR-C or parental sequencing data. And generating and analyzing all 3 separate data types is both costly and time-consuming. So there would be a clear and immediate advantage for many people working on human genetics to be able to do T2T assemblies with just 2 data types, merging 2 of these together, and the obvious candidates here being merging the nearly perfect and the ultra-long reads. In other words, what if we had near-perfect ultra-long reads? Now, unfortunately, we can't currently make ultra-long duplex libraries, because ultralong requires the transposase enzyme, and our duplex libraries require our ligation enzymes. So as soon as we heard that we could possibly get close to Q30 with simplex reads, we got very excited, and we wondered, are we now able to make ultra-long libraries that are accurate enough to be used as these nearly perfect long reads in the T2T recipe? And so we got a hold of an early version of this experimental condition and generated an ultra-long library with native HG002 DNA. In total, we sequenced 125 gigabases across all the flow cells, and the library read in 50 was 92 kilobases for this data set. Now, we got up to 99.81% median accuracy here. That's Q27, a little bit below the Q28 Lockmull just quoted, but like I said, this was an early version of the test. We now think we could get up to Q28 ultra-longs. We just haven't had time to generate that data set. But in this data set, we did get a 1.1 megabase long Q30 read, the longest Q30 read I think anybody has ever seen. It was HSAT2, so a low complexity repeat, but we even got an 839 kilobase pair long read that mapped to genic sequence, covering 2 long non-coding RNAs as well as 2 protein coding genes. Like with all Nanopore libraries, we also saw in this data set that accuracy was completely independent of read length. We got that beautiful Q27 median accuracy all the way out into our megabase long reads. So the question became, is this good enough now? Did we cross that threshold where the accuracy of these simplex ultra-long reads was high enough that we could use them to generate a T2T assembly without any external duplex sequencing? So all we had time to do, because this data literally rolled off the sequencers just over a week ago, was run it through existing off-the-shelf T2T assembly pipelines. And we chose the Hi-Fi ASM assembler with the Raft adjustment program. So Raft is a new program just released that allows Hi-Fi ASM to better deal with variably length reads. And so we ran it through this pipeline with minimal parameter tuning, no algorithmic adjustments whatsoever, and we got beautiful T2T genome assembly. So in total, we assembled a 3.03 gigabase maternal genome and a 2.94 gigabase maternal genome, both with scaffold N50s north of 130 megabases. We ended up with 19 telomere to telomere chromosomes, including 9 completely gapless contigs from one telomere all the way to the next telomere, and 10 additional telomere to telomere scaffolds. Again, I want to stress that this was using just off-the-shelf assembly pipeline, and we really hope, now that we are getting this data out into the hands of developers, that further optimization in these tools will allow us to see even better results as these tools learn to better handle longer reads with a slightly different error profile than what they've been used to. However, these results are completely consistent with what we see with similar depths of duplex or hi-fi reads combined with ultra-long reads and parental sequencing data. So right now, with just one ultra-long data set combined with parental sequencing data, we were able to generate T2T assemblies comparable to what we used to get with 3 different data types. And with that, I will turn it back over to Lockmull to talk about the assembly accuracy, though, and some fun, exciting updates in terms of consensus accuracy.
Thank you, Sean. So that's very exciting, isn't it? So that's just, I want to highlight again. So that is just simplex only, ONT only, telomere to telomere accuracy with high accuracy. So that's really exciting. So I have one more thing. So this is homopolymer accuracy. So for a long time, we've been talking about homopolymer accuracy. It is, even though accuracy, general accuracy improved significantly over the years, simplex Q20, duplex Q30, homopolymer accuracy remains still the largest mode of errors in nanopore sequencing. So we have discussed different methods to improve homopolymer accuracy further. We have actually, from when we moved from R9 to R10, homopolymer accuracy did improve significantly. In that pore change from R9 to R10, we increased the read head length. And we were talking about increasing the read head length even further. We are still working on that, but that project, we found out that it's very challenging. So we are making great progress, but still we are working on it. Because homopolymer accuracy is low, even though in nanopore sequencing you get very high SNPs and SVs, indel accuracy is not where we want to be. So last year in London Calling, Clive talked about 5B4. So in 5B4, what we try to do is changing changing the homopolymer accuracy by breaking the homopolymer with different bases. So with each base A, T, C, G, we have identified a special modified base that you can use. And when you try to make a DNA template with that modified base, you can see that it breaks the signal. So in black, what you see is the canonical base signal. And when you try to make another template strand with modified base, you can see that the flatness of the signal changes, which means there's more information that you can extract, and that will give you some information about the sequence. So for the time being, we pick just one base to modify, in this case T. So A, you can do with a simple fill-in experiment. You can convert to T, and when you make that T strand, you can, with the 4 canonical bases, you can include a modified T base, and then you can get 5 base strand. So this sample prep is not that difficult. So it's just a couple of hours. So as you can see on the right-hand side panel, so you have your DNA of interest, and then you add you do a PCR adapter ligation, and with that you add a primer and do a single filling step with 4 canonical bases and 1 modified base. And then you get your new DNA strands with modified bases incorporated, and that then we add rapid sequencing adapters, and then that library you can load into the flow cell to sequence. So when we do that, as you can see in this plot, the indel accuracy F1 score improved significantly. Even at low coverage, you can see at 30x coverage, indel F1 score improved from 87% to 95%. And at high coverage, at 90x, indel F score now reaches 99%. So this is a huge improvement. Actually, we were surprised to see that improvement. And with that, you can then, with this 5B4, if you polish the assembly, you can hit Q50 assembly accuracy. So this is, once again, a significant improvement to our accuracy. So which means we can now generate ONT-only T2T assemblies with high indels and very high consensus accuracy of about Q50. So just to summarize the DNA part, so in development, we have E8.2.1, and we will release that in Q1, seamlessly integrated into our existing kit. With that, you will get consistently Q20 plus accuracy. As I explained, we have a homopolymer fix with 5B4. We will release that in Q1 as well. In research, we are still working on to improve Q20 accuracy, Q28 accuracy I showed, to get Q30 simplex accuracy. And with that, we should be able to do nanopore only T2T assemblies with simplex with very high consensus accuracy as well. So again, this is the highlight of the talk. if you forget about everything else I said, just go home with this simple message. So we have Simplex Q20 accuracy. We have Simplex Q28 accuracy coming. And we have very high Q50 Nanopore only assemblies. And this is Nanopore only T2T assembly with high accuracy Simplex. So thank you very much. I think that's done. So next up is Rosemary Doko. So she will talk to you about our current product pipeline, timelines, and how are we planning to get everything I talked to you about to you guys. Thank you.
Thank you, Lakmal. Well, Lakmal and the rest of the R&D team make sure that our job is never boring. So my job is actually to bring you everyone back to earth. We're going to rush through our product range. But I'm also going to introduce you to our sample-to-answer workflows, which is something that we're also doing in R&D, apart from all of this amazing core platform science. So we have our product sample prep, sequencing, analysis, and just going into our sample preparation kits. Many of you know we've got a really broad range that enables you to do all of the amazing applications that you talk about on stage and that you publish in your papers. So released in shop today was the final tranche of our Q20 kits. So the 16S sequencing kit, the cDNA sequencing kit. We also released the single-cell protocol, moving that from a cDNA to ligation-based. So everything is now Q20 enabled. So kit 14 rollout complete. So if we look at our product range going into 2024, we have all of the flow cells for Flongol, Minine, PromethION, the R1041s are all released there. We've got the RNA flow cells, we've got the high Duplex in the registration-based early access, and all of the kits now available to order. So we're feeling really, really good about waking up on January 1st and having the full suite of products there for you. Lakmal mentioned E8.2.1. We're really creative about our namings in R&D, but it's actually really important to keep the convention simple. As he said, it is an incredibly minor tweak. So it's same production methods, the same concentration, same fill volumes, which means it's the same sample prep, it's the same software that runs it, the same base calling model, and we've also taken it through all the variant detectors like Claire3 and Sniffles and all the rest, and it's everything absolutely the same. So we're going to be rolling this out through Q1, starting off with things like ultra-long kits and rapid barcoding kits. That's where we see the biggest lift. And that's really, you can see, you'll be noticing a batch code change there. And it's really important for us because what we really want to do is get Q20 capability rolled into Q-Line, ready for Q-Line. The number of applications that come to us every day around what people are doing with the technology, what they really want to use the Q20 product for, we're really wanting to lock this down and get everyone robustly into Q20. You'll notice that we've started to use median because there's an ongoing debate about modal versus median. So that's fine. Regarding high Duplex, we continue to expand our registration-based early access. So register your interest, speak to the team, and then you get added into the high Duplex store. We are increasing the support. So later this week, Minnow will go out, and it'll be Duplex included. We also have new protocols and new kit components to support people wanting to do high Duplex. And so, of course, you're still exploring telomere to telomere, the hemimethylation and more metagenomic assemblies, and all the methylation support is now in Dorado. We've spent a lot of time this year making sure that we're accelerating our base callers, making sure that we're making the most out of all of the exciting upgrades from the compute industry and all the new silicon. So Minines can keep up on basic laptops, Gridines can keep up with Q20, PromethIONs can keep up, and we've been saying for a while that there's a little bit more speed to come out of that. And with the release of Dorado last night, that is now here. So we've got a 20% further speed increase on hack. That takes us up to the 48 flow cells for the PromethION 48. And that will be rolling into Minnow again during Q1. So now that we've got the speed piece done, we're starting to look at not just raw read metrics, but also other metrics around accuracy. So in September, we previewed a base caller model that improved our performance in bacteria. That's all rolled into the latest Dorado. And also the team have been looking at really, really challenging areas of the genome. So looking at improving the performance that we have in telomeres and centromeres, in low complexity regions, so things like alpha satellite regions where the algorithms have struggled before. And this is to get ever more complete genomes. And I hear that one of you guys who was at the workshop last night, you went home, downloaded Dorado, you ran it through, and you found the result that you needed. And that's fantastic. Hopefully everyone will be doing that soon, as soon as it gets into Minnow. But this is what the teams are really focused on, is really starting to get every single nook and cranny of the genome coming off the machines in production mode. So our algorithms have improved, the accuracy has improved, the compute industry has done an amazing job of bringing out new silicon, and it means that the little Mark 1C that's been our pride and joy, you see it on the front cover of every single thing that we do. It's time to say farewell. It can't keep up. But we will be, of course, supporting everyone who has one until the end of 2026. But you will start noticing that in the new year, we'll start pushing it to the legacy tab, particularly with the Mark 1D Min9 coming. So please do keep your eye on the community, because we will be offering full support in the transition over the next couple of years. So that will leave us with the following range of devices. You'll have our small handhelds, be it the Min9 and the P2 Solo. The benchtop ones, so the GridIRONs, the P2Is, and the higher throughput devices, P24 and P48. And we're going to be really starting to really distinguish the amount of, or how we sort of interact with users, be they Min9 or P2 users, and how we support users who have the larger devices. So we're investing a lot more into our digital technology so that we can really offer amazing support to the Min9 and P2 Solo users. But also, as you heard from Julie earlier this morning, we're investing a lot in our field application specialists and our sales team so that we can better support users with benchtop devices and larger projects. Moving on to Epitome, we've invested a lot in this platform this year, and it's really exciting to see all the feedback that we're getting. So we've always been really passionate about the fact that everything that goes into our informatics pipelines is open. So everything that gets packaged into Epitome, it is in GitHub, so you can pore over it, you can scrutinize it, you can understand what's going on under the hood. And then we package that up. And it's got intuitive interface, fantastic reports, you can run it in cloud, you can run it locally. And the really core workflow for us in Epitome is the human variation workflow. We've actually paired this also with lab protocols. So there's an end-to-end lab protocol where you run... Oh. So you run one simple protocol, one flow cell, and you also get automated Epitome analysis. So for 30x coverage, you're getting modification accuracy, mod calling at 99.6%, SV at 96.1%, phasing, megabase scale phase blocks, and also SNVs at 99.7%. And so this is the real workhorse that we've got inside Epitome at the moment for anyone doing human genomics. Producing variants is one thing, interpretation is another. And some of you may have heard that we're partnering with interpretation providers. So all of these, so Fabric, Genix, Varsome, all of these partners are there, and what we're going to be doing is you're going to be sequencing, and the data automatically can get pushed into your platform of choice. We're going to run an early access on it in Q1, so if anyone's interested, please sign up. It is something that we're really, really excited about. So hopefully you can see that we've got all of the pieces that are coming together, be it sample prep, be it sequencing, being it analysis and interpretation, so that you can really generate really, really high impact data. And we have got a versatile platform. You can do anything with it. Even just for human genetics, for example, you could be doing genotyping, low pass, standard, assembly, T2T assembly. And so what we are working on internally is a set of sample-to-answer workflows, so end-to-end workflows, to help everyone navigate this field of infinitive options. So you'll see that more and more we're releasing end-to-end guided workflows. So for example, on synthetic biology and core tools, we have a plasmid workflow, the AAV, the amplicons, all of these workflows delivering on Nanopore unique features such as information-rich, such as rapid turnaround times, such as that affordability and accessibility. On the small genomes, we have pathogen sequencing, many different flavors of pathogen sequencing, bacterial assemblies, metagenomics, again, everything from a wet lab protocol all the way to an epitome informatics, so that you can generate results. And then of human, we've got the human variation workflow. TeloSeq came out late last week, and you heard about TeloSeq earlier today. And then we've got in development tumor normal end-to-end workflows as well. So moving on to the roadmap, as Lakmal said, it's time to figure out how we get all of this innovation out into the hands of our users. You saw this earlier today. We've had 10 years of constant innovation at Nanopore. And we do that really because the core sequencing technology is in our flow cells, in our consumables, in our sequencing kits, and it's in our software. And that means that you can really run and innovate really, really quickly. So you spoke about accuracy. You've got the Q20 improvement that we're bringing out on Kit 14 and the 5B4 chemistry. But we also don't forget about robustness of output. And that's been a really big focus for us in Oxford Nanopore for the whole of 2023. So of course, we're driving towards 100 gigs in field, making sure that we're monitoring that, making sure that we're interacting with users as soon as if there's something that we can help them with in order to achieve that data output. But we also have more and more performance in the pipeline. And this always goes back to that fundamental value that we've always spoken about, which is when you're buying Oxford Nanopore, you're buying into the journey. You're not buying a fixed box that has a fixed set of capabilities. It will evolve. And particularly with PromethION, with some of the chemistries that the R&D team can access, particularly now with the advancement of machine learning techniques that we've seen Lakmal talk about, we will have the capability to be running 2, 3, and 4 genomes per flow cell, increasing the capacity of your device and decreasing the cost per genome. So we do also, as well as focusing a lot on all of the core platform innovations, we do spend an enormous amount of our energy figuring out how we're going to take all of that, freeze it down, and make it and get it ready so that you can develop assays on our platform. Now, Q-Line, I could go on for about 40 minutes about Q-Line, but I guess some people want some dinner. Yeah, look, the machine learning guy's going, yes. But actually, what we are going to do at London Calling next year, we're actually going to have a dedicated session to Q-Line, because there's actually a really exciting pipeline building up around it. But everything else that we do in our life science research tools, we go from beta to registration-based early access, open access released, freeze it down, and then we also have the partner products that we're working on. One of the things that's going to really accelerate Nanopore into applied markets is Turbots. And we've had one today on stand. I'm really impressed about how interesting you guys find robots. I've never seen as many people stare at a machine with moving parts, but it's quite impressive. It's been popular at every show, and the beta testing is starting now, and we're working with TCAN, who are a fantastic partner, and we're really, really excited about what this is going to do for us next year. So this is the usual homework slide that we get. So on our hardware, we've got our Turbot beta testing that we've announced today. We've got P2 Solo going from early access to released early in the new year. The P2i early access. The MinION Mark 1D will start beta testing as well in Q1, and then going into early access later in the year. On our platform side, high Duplex, that continues. And then we've also got the Kit 14 enzyme uprev that we've spoken about that will start flowing through in January. We have our Simplex, our high accuracy Simplex in 5B4. We'll be reaching out to some users and putting up some registration of interest forms so that we can start some beta testing. Importantly, Chris Wright did stay up late, and he has released Sean's data. So anyone who is a pure play tool developer can actually get their hands on data tonight. And the really important thing for us as well is to move Kit 14 into fully released mode so that we can start accelerating it into Q-Line. On the software and algorithms, it's been a busy couple of days for the team. So Dorados are out with the newer models, with more mods. Duplex support's going to go into Minnow this week. And then we're going to upgrade our cloud platform behind Epitome, tertiary analysis in Epitome, and then we're looking forward to integrating Epitome with Minnow in the new year as well. And then on the end-to-end workflows, we've got a number of things that we're really excited about that we're working on. And of course, we're getting a couple of upgrades for Q-Line at the end of Q1 with admin control, and then towards the end of Q2 and sort of later in that year to get the Q20 chemistry on it. So in this session, we only covered a fraction of what we're doing at R&D. There's a lot more, and it will be all shared with you at London Calling next year.